All right, welcome everyone for our Build a Cell seminar. Thank you so much, Angel, for showing up today. You're going to share a lot of your really amazing research with us, and I will turn the ball over to you to introduce the rest of yourself and then take us on a deep dive into what you've been working on.
Thank you very much, Carlis and Kate, for the invitation. So I'm going to talk about the intersection of computing and molecular biology and synthetic biology, and from the point of view of theoretical computer science. Okay, so I'm not going to talk about simulations or mathematical models, but I'm going to try to make a call about the importance of this intersection between theoretical computer science and living systems. So let me introduce my laboratory. I am Angel Goñi-Moreno. I am a computer scientist by training, but I have been for long involved in molecular biology. So I don't know if someone asks now, should I say I'm a synthetic biologist or biotechnologist or something like that? Okay. So this is the CBGP, the Center for Biotechnology, that belongs both to UPM, the Technical University of Madrid in Spain, and the CSIC, which is the Research Council of here in Spain. So this on the left, this is the CBGP, this is all staff of the CBGP. And this is my group. We are the Biocomputation Laboratory. So you are very welcome to take a look at the Biocomputation lab.com website and get in touch. So a big part of our activity is involved in dealing with tools. So we develop new tools to work with synthetic biology in a broad sense. Okay, so for instance, we are very involved in the standardization of components, of tools. We work on the synthetic with the synthetic biology language, developing, as for instance, this tool here on the left, Geneta, that turns genetic designs into networks. So you can then browse different networks and query designs using graph theory tools and so on. And here on the right, you see this SEVA illustration. We are heavily involved in SEVA, which is the Standard European Vector Architecture, which is a set of vectors, plasmid vectors, that have a very specific structure, okay? And modules that you can just cut and paste, different origins of replication, markers and cargos, and so on. Most recently, we have developed these PBLAM vectors, which of course follow the SEVA standard, and they are used to insert things into the chromosome of bacteria, okay? And the good thing is that this insertion benefits somehow the transposon sequencing afterwards, okay? So to be used in high throughput workflows. So continuing with the tooling, we are also heavily involved in laboratory automation. Okay, so far we have recently launched this website, which is the lab repo, the lab repository and format, which is just a database of different protocols to be used in the OT2 Open Drones robot. Okay, the protocols that are there now, they are for the OT2, okay, but more protocols will follow for different type of liquid handling robots. And what we did here is to define a format, as we did in the standard European vector architecture with the plasmid vectors. We define a format for the protocol, so all protocols follow the same format, and you can exchange classes and functions between different protocols in order to create a new protocol. Okay, so we are involved in all this tooling that goes along all the different steps of the typical synthetic biology life cycle: design, build, test, learn, and so on. Mathematical models as well, of course, are part of our tools. We have mathematical models for quite a few different mechanistic details of living cells. Here, for instance, in this piece, I'm not going to talk about this, okay? This is just to give you a sense of how, to what extent the mathematical models that we have explain different performances and behaviors. Here, what we did is to explore the role of dimerization, okay, when two monomers dimerize. Most specifically, we tackled the problem of what happens if two ribosomes are very close together translating the RNA, so then the monomers have a chance to dimerize on the go, okay? This little mechanistic dynamic is actually in charge of changing the performance of the genetic circuits by quite a lot, okay? So we have a lot of different mathematical models, all the code is open source, equations, of course, published and that throw quite interesting predictions. Not all of them has been already validated because, of course, we work at a different speed regarding mathematical modeling and experimental validation. Mathematical models, in this case, goes a little bit faster. So that regarding the tooling, like an overview of our laboratory, what is the biocomputation laboratory and what we do. And now I'm going to dive in the question that I wanted to talk about during this seminar. Okay, the previous was a kind of an introduction. So our fundamental question here is, can we design and build information processing, and not only information processing, but also computing devices with living material? Okay, so that is the principal question. So this is a Turing machine. I hope, and well, I guess most of the people here in the audience already know what a Turing machine is, okay? Well, not. Let me rephrase this. Most people here in the audience know that a Turing machine exists, okay? The notion of a Turing machine. Not everyone would know how a Turing machine works, okay? So just a quick word about the Turing machine. This is a Turing machine. A Turing machine is not a machine that you have on top of the table, okay? So not a hard machine with screws and that. Turing machine is a mathematical abstraction of how information at the input can be turned into outputs according to different rules. Okay, that's what Turing called a computing machine. Okay, and a Turing machine has three different things. A tape is this tape here in the cartoon with ones and zeros, but it doesn't have to be ones and zeros. It can be whatever type of information. That's number one. Number two is a set of rules. Here, you see the rules over there, okay, in this upper left. And a head. The head is this robot here, okay? Again, this is a cartoon. Okay? And the head is in charge of reading the tape and writing in the tape according to the rules. So in this case, the machine just stopped because the head is reading a 1, and it was in state A. So if you go to the rules and you say state A, you are reading a 1, then you write a 1 and stop. I don't know why the cartoon has this skull here, but this doesn't mean that the machine explodes or something, okay? This is that machine stops. So that's the Turing machine, and that's a very elegant design because and very powerful because our understanding of computation still is this thing. Okay, this is the understanding, the current understanding, like 70 years ago or something, of computing is that of a Turing machine. So when we look at a living cell, and this is the typical cartoon, from what cell. When we take a look at a living cell, we see a lot of information, for sure, information that is coming in from outside, information that is inside in different ways. We see information that is being encoded into DNA, and we see different rules. Transcription is a rule, translation is another rule. The fact that transcription needs to come before translation is another rule, and so on and so forth. No? So, okay, so there might be a possibility, and that's a core question of our laboratory. There might be a possibility that we find here ways of playing with information, of turning inputs into outputs according to different rules that go beyond what the Turing machine can do. Maybe not just beyond, maybe alongside or something like that, but out of Turing. So looking for a non-Turing computing, that's at the very core of our laboratory. So in this regard, a few years ago, 2019, so 4 years now, we proposed to use this notion of cellular supremacy, okay? In the same way as the quantum computing people are claiming that they are going to achieve this quantum supremacy, okay, that quantum computers are going to outperform classical computers at different tasks, we are claiming that we are going to achieve this cellular supremacy, okay, that cellular computers will also outperform classical computers at a specific task or in different domains. And we had this figure in this cellular supremacy paper that I like a lot, and it shows how, conventional computers and biology, okay, biological systems, have very different, but also this type of rules and instruction sets that turns inputs into outputs. No? In computer science, you find combinatorial logic, and then finite state machines, Turing machines, all in increasing complexity. Okay, with combinatorial logic, you cannot do that much. These are relatively easy and very weak model of computation, and then finite state machines, Turing machines, and so on. And in biology, you have very different processes, let's say, or mechanistic primitives, also we could say, that are used to process information at different levels. So gene regulation, we all know about gene regulation and how gene regulation can perform different functions. Metabolism, of course, is very different from gene regulation, and it's also used by cells to deal with information in a different way than gene regulation. We have processes such as evolution, which is somehow a model of computation in itself, because evolution deals with problems for adaptability and so on. No? So there is a problem, there is a question about how to adapt a living system to an environment, and finally there is a solution. So we focused a lot on combinatorial logic. I said it is one of the weakest model of computation, if not the weakest of them all, but it's very useful when trying to map the Boolean logic functions to gene regulatory circuits. So we work a lot with genetic combinatorial logic. I guess some or most of the people here in the audience are familiar with this, but I'm going to explain it very quickly. So combinatorial logic means that a specific combination of inputs is going to produce a specific combination in the output of a circuit, for instance, okay? Here in this picture, we have a piece of DNA that is producing a protein, okay? A transcription factor. We can introduce some regulator that is going to inhibit this performance. And this is a perfect 0-1 relationship, okay? If this regulator is not present, then we have an output, and the other way around. If the regulator is present, this is repressed, and we do not have an output. But we can extend this more, and we can extend it, and more, okay? And at the end of all this circuit engineering, what you have is a large, to some extent, piece of genetic circuit that reads some combination of inputs and delivers some combination of outputs. So we are doing this with our favorite workhorse, which is Pseudomonas putida. Okay, this is a gram-negative bacteria. Why Pseudomonas putida? Okay, years ago I really didn't care about what cell I was going to use, because as a computer scientist, what I wanted to do is these computing devices, and I didn't care about the chassis at all. Now I do care a lot, because the applications that I am going to deal with, that I am interested in, are applications in the environment, okay? Environmental applications. And Pseudomonas putida happens to be that it's an environmental microbe that is already doing a lot of interesting functions in soil, like bioremediation of toluenes and so on and so forth, that are very interesting from the point of view of applications of genetic regulatory circuits. So in this case, this is a NOR logic gate the NOR logic gate works as follows. This is the little simple symbol of a NOR logic gate. NOR logic gate has 2 inputs, 1 output, and it performs according to this set of rules. Okay, only when both inputs are 0, the output is going to be on. In any other case, the output is going to be off. So this is the simulation, and we assembled this circuit here above. And these are the experimental validation with 2 slightly different circuits, okay? And the circuit performs as follows. We tried to do this with parts, genetic parts. When I say genetic parts, I mean this promoter here, this gene here, this repression from here to here, blah, blah, blah. We try to do this with parts that were very well characterized in E. coli, which is another cell. And we thought, okay, well, I would say I thought, I don't want to blame any other one than me about this, but I thought that, well, if it worked for E. coli, it should work for Pseudomonas putidi, and that's a big mistake, okay? As somebody said, what is true for E. coli doesn't have to be true for an elephant. Okay? So, okay, we came here to this picture, okay, or something similar, okay, the concept of this picture, and say, okay, genetic circuits are just a small, tiny bit of what the components that we have in a cell. So there has to be, of course, some interactions between the genetic background, what we call the context, and our circuit. And from that moment, we started exploring these specific interactions, this specific dependency, which is the name that we assign to this issue, right? The contextual dependencies of a genetic circuit. And that is a very simple concept, right? When you measure a genetic a cell with a synthetic regulatory circuit, and this is, I guess that you see the cursor, let me put this laser or whatever. Okay, when you measure your cells, and this is a typical graph, this is a transfer function that you get, you are not measuring only the genetic circuit, okay? Although we would love to be measuring only the genetic circuit. That is not the case. In reality, what you are measuring is the intersection of your genetic circuit with the plasmid that you are using to have this genetic circuit in, and the interactions of these plasmids and the host organism that you are using. So what we did is an interesting study of taking 20 genetic inverters. Okay, this is the inverter. All this circuit is a NOT logic gate, an inverter. We took 20 of them, and we did a lot of combinations, okay, with different plasmids and different hosts. What we had at the end, I think, is a collection of like 140 different functions or something like that. Okay? So from 20, we had 140 different combinations of gate context combinations. And we started exploring what was the performance of the whole library and trying to get some conclusions of that performance. Conclusion number 1, which is kind of disappointing, okay, but that's what it is. The relationships between one context and another context are not linear, okay? There are non-linearities everywhere. Of course, this is a complex system, both the genetic aid and the interactions between hosts and so on, and the non-linearities are everywhere. So that makes very difficult to predict, but an advantage is that it makes it easy to reuse. So now we don't have 20 different inverters that we can use. We have 140 different inverters that we can use, all with different functions. So depending on the function that you want, you can choose the inverter and the context that you want your inverter in. So an example of the non-linearities is here in these pictures on the graphs in the right, and I'm going to show you with a couple of examples here, okay? So if you take a look at this context, here, this is the measurement of 2 genetic NOT logic gates, which are the red and the green one, okay? So, okay, this is in, the 2 gates in one specific context. Now, if you change to this other context, to the right. Okay, there is a chat message. Okay, this is a question. I will deal with that later. If there is a pressing concept like I don't know, we can hear you or something like that, please speak up. So if we change from this context to this other context, what we see is that the two gates change the performance in a very similar way. Okay, that's a success story, let's say, because if this stands, that means that we can write like a translator from this context to that context. So everything here in this context would be automatically translated into that other context. But then take a look at this context here in the top right. The two gates change their performance, but in a completely different way. Okay? So the dependencies between the background and between the context and the circuit are nonlinear, and that makes predictions being quite a headache. But as I said, the compatibility between inverters increases by a lot. Okay, so in synthetic biology, you need to put several logic gates in a row for whatever the application, and if that is the case, it happens to be that one gate needs to be compatible with the following one in the chain. Okay. And that's a challenge to find compatible gates that can be assembled together. So here, if you see this graph here on the left, you see that the compatible pairs between different gates in this library, the dark blue squares, are not that many if we are using one plasmid only. So we reduce the number of combinations to only the genetic circuits. Okay. But if you allow for all the plasmids to be used, you increase the combinations, and within all these combinations, of course, there are more compatible pairs. If you allow for all different contextual combinations, so you are using the full library, there are a lot more compatible pairs. So that's a message that I want to stress, which is the importance of the context. You can design with the context. Given a function, if you don't like that function, you might as well try to fine-tune the context instead of saying, okay, let's throw this genetic gate to the bin and build another one because it doesn't work. Okay, so the context can be used to design and to fine-tune. We are extending this more and more. Here, for instance, we are adding Pseudomonas protegens to the game. Okay, before we were using 2 strains of E. coli and 1 Pseudomonas putidum, and now we are using Pseudomonas protegens, so 2 different Pseudomonas. And we have a very interesting fact, that is that both Pseudomonas, here is the PF5 and the KT2440, so the blue line and the purple line, both Pseudomonas behave quite close in comparison to the 2 E. colis that we are using in most of the examples. But in some of them, it is not the case. Okay, you see here this graph, and 1 E. coli, 1 Pseudomonas are very close in performance, and the other E. coli, the other Pseudomonas are very close in performance. So again, a little pain in the neck, okay? Because if it were the case that all Pseudomonas behave more or less in the same way, then we could have concluded some kind of context rules or something like that, but it wasn't the case. So we will keep looking. So this is the typical slide that I always use to refer to this contextual dependencies work, okay, which are so a couple of pictures from this contextual dependency hell that we call in computer science. Okay? Here in the right, you have these Python libraries intersecting with everything in the OS of your machine. So for instance, if you have a code in Python that works very well in your machine and you share it with another machine, a different OS, maybe Linux versus Windows or whatever, and most probably the code will not just work at least equally well, okay, because it interfaces with the background of the machine, with the context of the machine in different ways. So this is the dependency here, and I like a lot this picture here on the left, because it tells a story about these contextual dependencies very nicely, right? the pieces of this Tetris game are perfectly fine. There is nothing wrong with these green, blue, and gray pieces. No, they are fine. You should be able to play with them, but you need to know what is the world in which you are playing in. Okay, this means that okay, your genetic circuit might be as precise as you want, but you need to be aware of the host that you are playing in. So we are looking at quite a few different contextual dependency stories, sub-stories, let's say. One of the sub-stories is a space. Okay, bacterial cells are small, of course, okay, but space within the volume of the cell matters quite a lot. Okay, we had this work published a few years ago in which we had some constructs. The constructs were genetically the same, but it was divided, the circuit was divided in two. Take a look at this picture here, okay, in the this flow cytometry graph. The circuit was divided in two, and in one experiment, the two pieces of the circuit were very close together, and in the other experiment, the two pieces of the circuit were very far apart. Okay? The performance was different. That was interesting because the genetic circuit is the same, the sequence is the same, the host is the same, but the performance was very different, okay? Because there is this little contextual dependency feature that you can play with, which is a space. It actually matters, the location within the chromosome, for instance, that the pieces have been inserted in. Another contextual dependency that we like a lot in our laboratory is noise. Okay, we like noise a lot. This is one of our images. It tells what is this gene noise, genetic noise, very easily, right? All these cells are expressing GFP constitutively, but some of the cells are brighter than others. Okay, of course, the genetic signal, molecular signals are not digital. This is not 1 and 0 on or off, but this is analog. Okay, and this is one of the features that we actually see as promising to achieve the cellular supremacy, because electric engineers are quite, I wouldn't say, I don't want to say they are bad at, but electrical technology, let's say conventional electrical technology, is not very good at processing analog signals, but living systems are ideal. Okay, they have been working with analog signaling for millions of years, and it's an evolutionary advantage. So we should be looking into analog processing instead of digital Boolean type of processes. Having said that, analog signaling is to some extent a problem if you are trying to build Boolean networks. So we have this little trick in order to try to digitalize a signal, which is what we call the digitalizer. Okay, the digitalizer is this module here in which you can place your gene of interest, or you can place your promoter of interest, and all of a sudden the promoter is going to give a 0-1 type of signal instead of a very leaky 0 signal and a very low 1 signal, and so on and so forth. This has a double negative feedback loop that I'm not going to get into details unless you like. But what the message that I want to send with this is, it is true that analog signaling is quite important, and we have identified analog type of computing, probabilistic computing, and all that stuff as a pathway to cellular supremacy. But if you want to play with digital type of signals, for instance, you are working with a heavy toxin or something like that, then we have a problem. We have a solution for it. So, okay, so there are contextual dependencies, these noise problems with different cells, problems with space, and so on. So at some point, we said, okay, what if we are designing the circuits wrong? Okay, So then this is a story about the structure, and this is going to be the final story here of my talk, and then we can discuss about the different topics. But this is the latest, one of the latest stories from our laboratory, and one that I like a lot, and is well, querying about what happens if the intuitive design of genetic logic gates is actually not ideal. Okay, so for instance, this is an example, the toll network. This is a metabolic and regulatory network of Pseudomonas putida to degrade different pollutants and different aromatics. This toll network that is so complicated as this, okay, and this is already a simplified version of the full network. This can be abstracted away into an OR design. You can see here the OR design. You have two inputs and an output. But the actual structure of the network is not what you would design if you had to build an OR gate. It's much more complex, okay? There are redundancies, there are connections that are apparently doing nothing. So maybe we can approach that weirdness, let's say, in the architecture of the networks in order to see what happens. What we did is as follows. So we took an AND logic gate as a target function. Okay, this is an AND logic gate. And we said, okay, how would you implement this AND logic gate if you were a human, what we are, okay? So this is here above the intuitive design of an AND logic gate with the pieces that we have in our laboratory. Okay, you have the two inputs and you have the output, and there is a linear forward process of information. The information always goes from one step to the following, one to the following, one to the following. This is the typical way you would do an electronic logic circuit. So this is the simulation of the AND logic gate, and you see the green line be the highest, okay? Because that's how the AND logic gate should work when both inputs are one, the green, then the output is high, okay? What we did is we implemented a genetic algorithm, and we fed the genetic algorithm with all these parts that we had in our laboratory, all these PTAC, PTET, and so on. Okay, and we said to the genetic algorithm, okay, so this is the function, the graph on the left, that you have to optimize. We need as high output as possible. And we do not care about the topology of the network. We do not care about how many times you use the same promoter, how many times you go back, the information goes back a step. If it goes forward afterwards, we do not care about redundancies and so on. And after 64 generations of our algorithm, this is the topology that the algorithm found. Okay, you see it here in the top right. Okay, and this network outperforms the one that was the intuitive design by quite a lot. Okay, you see that by generation 416, the network doesn't even behave as an AND logic function. Okay, the green was low and so on. But after a few generations, you see generation 64, you have a very, very strong correlation between the two inputs and the AND and the high output. And this actually outperforms the simulations. Okay, this is a simulation. We are testing the experiment at the minute. So maybe we have another talk, I don't know, in a few months, that we'll be able to tell you what happened with these designs. But in the simulation, the network in the generation 64 already outperforms the traditional one, let's say, by orders of magnitude. It is very nice because this network here, you wouldn't be able to tell me what that function is if I ask you and give you like an hour or something, no? If I tell you, okay, well, maybe an hour to come, no? But it is not easy to say what happens if these are two inputs and this is the output. What type of function is this? Woof, yeah, you would have a hard time, okay, taking a look at all these connections and repressions that are linking this and that, and again, redundancies and so on. So we are now building this because it might actually happen that the intuitive designs that we are using for Boolean logic circuits are not even the proper ones. So that's basically it. No, so I wanted to stress the fact that the Turing machine and all these concepts about Boolean combinatorial logic, finite state machines, memories, and so on, all this belongs to theoretical computer science, are just theoretical ways, mathematical ways of formalizing how information is being processed. The fact that you are using conventional technology in order to build a conventional computer, that doesn't mean that you could use different types of technology, like living technology, to build the same abstracted models of computation, which is what we are doing. So the take-home message of this talk is that biological systems compute, and that sounds like trivial to me, at least, but there is a quite a passionate debate about this sentence. But I am I tend to say that biological systems compute. By using living matter, we can build computing devices that may be outside Turing principles, as I said before. Evolution might have found different ways of processing information for sure. So we can even think about processing information in domains that are unreachable by conventional computers and achieving what we term the cellular supremacy. So that's the talk. This is a group, founders and so on. So, well, I'm happy to take any questions from no one.
Yes, thank you so much, Angel. I think that there are a couple questions in chat still. And for the people who are maybe watching the video at home, if you can read anything out loud that gets posted in the chat before you answer it, that would be awesome. And then a welcome to anybody to unmute themselves and ask questions as we go.
So should I okay, should I read the questions? Okay. So Daniel said, in your contextual dependency work, what was the highest number of genes you explored together? Do you think it would be possible to translate those findings to 200 genes? What we need for full synthetic cell? Okay, so that's difficult. Okay, we tried to minimize actually the number of genes that we were moving from a context to another context, because I really want to find general rules that tells you how that context interacts with that set of genes. Okay, so the logic gates that we're moving around are in the order of maybe 6-7 genes, no more than that. I have no idea what will happen if you move 200 genes. That to me sounds like a lot. I always play with very few genes. So the Python hell, okay, I think it's a very good metaphor. Yes, indeed. So Ana Maria says, does the gate performance change with the size of gene expressed? I'm not sure about the question of what gate. But in short, I could tell you that the gates change the performance with virtually everything. Okay, the slightest change, even the smallest change in you have two terminators instead of one, you have a slightly larger gene instead of a shorter gene, blah, blah, blah. Everything changes the performance of basically all the constructs, okay? So this is actually one of the most interesting things about playing with living matter, right? Because living systems have evolved to be specifically that thing they are now, okay? So if you change them a little bit, the performance is going to change, and the size of the genes matters.
They have, and I think you answered the question, I think, but they have added to that the logic gate that you tested in the contextual dependency work.
The larger one was, I think, the initial library of the 20 gates were all about the same size. So I don't think there is any correlation, at least in our data set, about the size of the gates.
Sorry, that was my bad. Logic gate.
The one we evolved with the evolutionary computing, with the genetic algorithm, or what? Okay.
I believe so.
So the one that we, the logic gate, the one that we evolved with the genetic algorithm, the one that was quite, yes, that was quite big. Okay. So we in that algorithm, in the evolutionary algorithm, we had a limit in the number of parts the algorithm could use, but the algorithm didn't select them all together. So there was a threshold. Okay, so the algorithm found the ideal gate up to what we thought. Okay, so like 9, 10 genes, something like that. Below that, beyond that, more genes were not making a better gate. Okay, so I don't know if that was because of the mathematical models that were behind the gates that we enter into the algorithm. It is a complex question actually, but there was that specific threshold beyond which the gate didn't work as well as with 9, 10 genes. Okay, but we will know more about this when we do the experimentation, of course. Do the gates behave similar? If you had 2 or more in the gate instead of single origins of replication, I guess. If you had one or more origins of replication within the gate instead of single gene gates. No. That's an interesting question. I don't know. Yeah, openly different. Yeah, the multiple genes. Multiple genes. It shouldn't matter actually, because what matters the most is the connections. Okay, so the interactions, the regulation of them. But whether you had more than different open reading frames, I don't think that being a different, or at least I cannot see how that would modify the performance, or in which sense. Okay. Okay, so I think I went through all this chat, but if someone can maybe speak up, that would be nice, because so far I've been talking to my computer and reading my computer for a while. So maybe there is a live question or something.
Yeah, but oh no, sorry. Just a lot of compliments in the chat. Yeah, we're not huge fans of live questions when we don't have to do them. Um, it was a wonderful talk though, and I'll put out that final call for questions. Of course, feel free to unmute yourselves or toss it in the chat. But thank you so much. And Hal will hover over this chat for a little bit before I stop the recording. But thank you so much. It's dense, complicated work. I'm excited. I'm going to have to rewatch the video and just kind of get deeper into it myself.
Okay, thank you very much. Thank you for the great words, everyone here in the chat. And I am very willing to engage in any other further communication if people want to get in touch.
Fantastic.